today. Who remembers where we left off on Wednesday?
We were
Passive transport.
about to look at active transport, I think.
Passive and active transport is where we were. So I wasn't quite done with passive transport yet. So we'll put off active transport for a few minutes. And we've got just a few things that we need to talk about with passive transport. So let's bring up our notes. Maybe. There we go. Okay. So passive transport. And there were 3 types of passive transport. Can someone remind me what those were?
Diffusion, facilitated diffusion, and osmosis.
Okay, so those are our 3. We need to talk about each one of these 3 to make sure you know what they are, okay? So, but before I do that, what if, let's say we've got a glass here, and it is full of water. And then I take some red food coloring and I drop a little drop of red food coloring into the water. What's going to happen? Georgia, what's going to happen when I drop that food coloring into the water? What do you think?
Well, if it's one drop, it probably won't do anything.
It won't be doing much. So let's add one more. Let's make it do something. Let's add 2 more just for fun. What's it going to do from there?
A very light pink, maybe.
So the whole water is going to turn a light pink, right? It's going to diffuse out to the rest of the water. And so let's see if I got pink. Do I have pink? Kind of. This is a kind of a pink. What we're going to get is this water that kind of looks pinkish, right? Let me get, oh, I just got rid of all my pink. Let's try that again. It's going to look something like this, okay? Except for it's going to be solid. I can't do solid with pen, at least not to my knowledge, okay? Water's going to look pink. It's going to diffuse out and fill the entire glass full of pink water, okay? It'll be a light shade of pink. You're totally right about that. But it's not, you're not going to see those big central concentrated drops of red out there anymore. So that's what diffusion is when things diffuse out and fill a space, okay? So typically what we think of when we think of diffusion, we think of, I'm going to use the word solutes. Who remembers what a solute is? I want to make sure you remember that word before I talk about it. Solute. What is a solute? It was like week 2.
A solute is something that like saturates, I think.
So solute is something that is dissolved in something else. So salt, if you put salt into water, it dissolves, right? Okay, it dissolves. And so a solute is something that dissolves into something else. We call whatever it dissolves in, like water, we call that a solvent, okay? A solvent is what dissolves something else like salt, and salt would be the solute. So diffusion is when solutes go, oops, not what I want. From an area of high concentration, I don't have enough room to write the whole word, high concentration to low concentration. Okay, low concentration. So just like with this little glass of water, when we dropped in just a few little drops of food coloring in it, it diffused out and filled up the whole area. So it was kind of like this homologous, clear pink mixture throughout the whole thing. Okay, that's what diffusion is. Went from an area of high concentration of red out to being a whole big area of being a big filled pink glass of water. Okay, that's diffusion. The other thing you need to remember about all three of these, actually, I should just put all, this is all three, a characteristic of passive transport is that no energy is required. So that's true for diffusion. True, okay? We go from an area of high concentration to an area of low concentration, okay? So the rest of the glass had low concentration of red food coloring, and therefore, where it was in high concentration, it diffused out to areas where it had lower concentrations, okay? Now, next one that I want to talk about is facilitated. FAS or FAC? I just forgot. Let me just see. It's FAS. FAC. Yes, FAC. Thank you. So facilitated diffusion. So diffusion is in the word, right? So we got diffusion in the word. So it's going to be this definition up here is going to work down here too for part of it. There's a little extra to it, but let's go ahead and put that in there just for fun. Okay. High to low concentration. Now, but here's the other part of this. This is the facilitated, and it requires a transport protein. Okay. So let's draw a picture so we all are on the same page. So remember our fluid mosaic model of a cell membrane looks something like this. Not making sure there's enough of these tails, but this is our cell membrane, look like something like this. These are phospholipids, the little guys here, and this big guy is a protein. Okay, and these proteins act as doorways, right? It is really hard for me to walk through a solid wall. I mean, statistically, it is possible, but it's astronomically high. So it's really difficult for me to walk through that wall. It is much easier for me to go through a door, right? Much easier for you too, I promise. And so for some things, it's just not feasible for it to pass through our membrane, okay? Like diffusion. Diffusion would be an example of something that just passes through the membrane. Facilitated diffusion, on the other hand, requires a doorway, and that would be these transport proteins, okay? These transport proteins, okay? Again, no energy is required. It's just going from an area of high concentration. So here's my solutes. These pluses are my solutes. High concentration. And they can pass through and go to an area of low concentration, okay? Okay, that's number 2. Not much different from regular diffusion, just requires a doorway in order for that to happen. So I want to talk about osmosis next. We all wish we could learn by osmosis. You've all wanted to put your head on your pillow and just learn by sucking that information in right in through your head. Unfortunately, for all of us, it doesn't work that way. At least not in the context that we think. Okay, so osmosis. Oops, what happened? Are you guys still there? My screen just went white.
Yes, I am.
Okay.
Yeah.
Let me just make sure I didn't lose something. You can still see my whiteboard?
Yes.
Okay. All right. Whew. That was weird. Okay. For me, apparently not for you guys. All right. So what is osmosis? This is a situation. Instead, up here, we're talking about these solutes, things that are dissolved in water. Okay. That's what's moving around. With osmosis, it's different. It's still diffusion, but it's where water is moving, okay? So water, okay? Water moves. I got a video I'm going to show you in a few minutes, so hopefully this will, I'll share it with you, and then I'll hopefully show you a video that will even make it even more clear. So we got a membrane, just like our cell membrane, okay? Our cell membrane lets some things through, other things can't get through. They need a transport protein or something in order to get through. Same with this membrane, okay? We call it a semi-permeable membrane. It's semi-permeable because only some things can get through. Usually water doesn't have a problem getting through our semi-permeable membrane, okay? So we've got water. And it can pass back and forth in this membrane, and it does, okay? Let's say we've got a little salt. See, let's draw a little salt shaker. It's got little dots in it. And lots of salt, and salt comes out and goes into our water. So we've got lots of dissolved salt particles over here. Now, if this is tap water, there is naturally a little bit of salt in our regular tap waters, but in very, very low concentrations, okay? So we'll put some of that over there. So which side has more, a higher concentration of salt? Over here, right? High concentration of salt. As a result, the water concentration is actually a little low in comparison with the other side. Water is high over here, high concentration of water over here, salt low, okay? And so water is always going to go towards the direction where there is a lower water concentration, okay? And because there's something else taking up space in the water, like salt, the lower water concentration is over here. So water is going to always head where there's lowest water concentration. So let's write that down. Water will move towards areas of low water concentration, okay? So typically, in order to figure out where you've got low water concentration, you look for places that have high solute concentration. So water concentration will be low where there are lots of solutes. Remember, solutes are things that are dissolved in water. So water, we'll label this side A and this side B. water is going to move in this direction. So in the end, we're going to get something that looks, I'll draw another picture over here. We're going to get a situation that looks like this. Okay, the water had moved over. Okay, so that's osmosis. Again, if you put lots of salt or something that dissolves in water, water's going to move towards that direction. Okay, we're going to watch a little video here to try and clarify that a little bit, and then we'll talk some more afterwards. Okay, so throughout the semester, I've put up a few videos by a YouTube group called the Amoeba Sisters. I like them because they use cartoons and I think they make it nice and simple and straightforward. And I'm always for simple and straightforward solutions. So I'm going to show this this video to you, and let's see how it works out for us here. I got to change screens here. Okay, here we go. Oh, no, wait. Wrong video, I think. Osmosis and water potential. Nope, never mind. This is the right video. Here we go.
When we were kids growing up in West Texas, our winters would be cold but rarely experience snow. But we did have ice, which resulted in the roads being salted. As the salt mixes in and dissolves into water on the road, this can lead to a lower freezing point, which can help prevent the roads from icing over. And while this is great for making the roads more safe, it wasn't so great for the plants that lived right along the roadside. It often caused them to die. Now, winter can be hard for many plant species, but I'm talking about this salt affecting even some hardy plant life. This issue with salt in plants isn't limited to winter. During hurricanes near the coast, salty ocean water can be dumped in large quantities into the soil. This can eventually kill plants, including trees that had originally survived the hurricane. Why? Do plants just dislike salt that much? Well, it's actually related to a term called osmosis. When you are talking about osmosis, you are talking about the movement of water through a semi-permeable membrane, like a cell membrane. Water molecules are so small that they can travel through the cell membrane unassisted, or they can travel in larger quantities through protein channels like aquaporins. The movement of water molecules traveling across a cell membrane is passive transport, which means it does not require energy. In osmosis, water molecules travel from areas of a high concentration of water molecules to a low concentration of water molecules. But there's another way to think about water movement in osmosis. A low water concentration likely means there is a greater solute concentration. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there is a higher solute concentration, which would mean less water concentration. So if you want to easily figure out where the water will travel in osmosis, look to the side where there is a greater solute concentration. Unless we bring in another variable, like pressure, water will generally have a net movement to the area of higher solute concentration. So let's bring out a U-tube. U-tube, that's funny. There's a semi-permeable membrane in the middle of it. Let's assume that it is similar to a cell membrane and that water molecules can squeeze through it. The molecules are quite small, but salt can't. Right now, there's just water in this U-tube. The water levels on side A and side B are equal. That doesn't mean that the water molecules aren't moving. Water molecules like to move, but the net movement across the two sides is zero. That means the overall change in direction of movement is zero. Now, let's imagine on side B, you dump a huge amount of salt there. So which direction will the water initially move towards? A or B? Think about what we mentioned with osmosis. The answer is B. Side B has a higher solute concentration than side A. Water moves to areas of higher solute concentration, which is also the area of lower water concentration. You will also see the water level on side B rise as the water moves to that area. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water across the two sides will be zero, but remember that water still likes to move and movement still occurs. Now here's some vocabulary to add in here. We call side B hypertonic. That means higher solute concentration. But we can't just say something is hypertonic without comparing it to something else. We say side B is hypertonic to side A because it has a higher solute concentration than side A. In osmosis, water moves to the hypertonic side. We say side A is hypotonic. Hypo rhymes with low, which helps me remember that it's the low solute concentration when compared to side B. Let's get a little more real life now instead of just the YouTube. As you know, water is important for your body and many processes that occur in the body. When someone gets an IV in a hospital, it might look like the fluid in the IV is just pure water. But it is certainly not pure water. That would be a disaster because of osmosis. Let's explain. Let's say hypothetically pure water was in an IV. Now an IV tube typically runs through a vein so that you have access to your bloodstream. Really useful for running medication through. Blood actually consists of many different types of components, and red blood cells are a great example. So what do you think has a higher solute concentration? The hypothetical pure water in this IV tube, or the red blood cells? Well,
What do you think? Is it the red blood cells or the pure water? Who's going to have more solute concentration?
The blood cells?
The blood cells, right. They're going to have salts. They're going to have all sorts of things dissolved in them, right? So absolutely.
cells are not empty vessels. They contain solutes. The pure water that hypothetically is running through this IV tube has no solutes. So where does the water go? It goes to the area of higher solute concentration, which in this case is inside the cells. The cells are hypertonic compared to the pure water in the IV tube because the cells have a greater solute concentration. The cells would swell and possibly burst. Exploding red blood cells are not good. If a person needs fluids, they typically will receive a solution that is isotonic to their blood plasma. Isotonic means equal concentration, so you won't have any swelling or shrinking red blood cells. Another example, let's talk about the aquarium. I have always wanted a saltwater fish tank ever since I was a little kid, but I've only had freshwater tanks.
Okay. They're going to go on to explain something similar with fish. Basically, if you put fresh water or pure water in with saltwater fish, their cells expand and explode because that's where water goes. Water goes into places with higher salt concentration. So totally not a good thing, right? Okay, we lost somebody. Who did we lose? Dang. Well, that's sad. Oh, they're back. Here we go. I think they're back. I think they're back. Ah, there we go. Okay. All right. Anyway, so let's bring up my notes again.
So far. I did often question when I was a kid, but we did have ice, which resulted in the roads being...
Stop. Okay. All right. Notes. Okay, so here's our notes. We learned something here. We learned something about the word hypertonic. And isotonic. Oh, sorry. We did learn that word, but that's not the word I want right there. Hypotonic. And we learned that water heads in the direction of hypertonic because hypertonic means high salt concentration. So high salt concentration or high solute concentration, sorry. And hypo means low solutes. Did anyone catch the word that meant equal solutes on either side of a membrane? Started with an I. Anyone catch that one? So isotonic means equal concentration. So in your blood cells right now, hopefully, unless something weird is going on in your body, all of your cells are in some kind of isotonic solution so that you don't have some weird osmosis thing going on. We don't want lots of water running into our cells. Otherwise, our cells would explode. Got a story for you. Any of you listen to the radio besides, Pandora or something like that on the web? You guys are too young for radio. I grew up in a time where if you wanted to listen to music or news, people turn on the car radio and listen to the radio most of the time. We didn't have iPhones, and we didn't have our Android devices that we have today to listen to music. So we listened to the radio. Everybody did. And the big thing was the morning radio show. Every radio station had their morning radio show, and they'd always do crazy things and crazy stunts. And one radio show in Minneapolis decided to do a crazy stunt. And you had to come down to a car dealership or something like that. And if you could do a gallon challenge full of water, then you would get like a free gaming system, like, I don't know, Nintendo or whatever it was.
A question.
Yeah, go for it.
Isn't it dangerous to sort of consume that much water?
It is very dangerous. And when you're going to hear what happened. So not only did they do a gallon challenge of water, but they used pure water, like distilled water, okay? Which is even more pure than just regular spring water, okay? So they, let me draw my fake gallon of water here. Here's my fake gallon of water. That looks terrible, but it is what it is. That's pure water. And then this lady comes down. Draw my terrible picture of a lady here, thinking she's going to get a really cool gaming system. She's got lots of solutes in her, lots of solutes in her body. And she drinks a gallon of water, keeps it down. That was part of the game. And so where's the water going to go? What's going to happen?
It's going to go throughout your entire body.
It's going to go. Yeah, go ahead.
Just diffuse all this, those solutes and stuff.
Yeah, so all that water is going to go right inside of her cells. So you remember the picture of the red blood cells? It's going to go into all of her cells, and many of them are going to burst. And that's exactly what happened. And she died later that day. How sad, huh? All for a radio competition. Someone didn't take their Bio 100 class in college. That's all I have to say. So no, a gallon challenge of water is very dangerous. No, absolutely don't want to do that. Thank you for pointing that out, Jamie. That is absolutely true. Yeah, we don't want that to happen. So was she hypertonic or hypotonic? Was she a hypertonic or hypotonic?
Hypotonic?
So the water is hypo, and she was hyper, okay? Because she's got all the solutes dissolved in her body, okay? Hyper, okay? Now, of course, we want isotonic, which is equal, okay? All right. Let me, in your textbook, what you're going to see is in a hypertonic solution, you're going to see a picture of a cell. Oops, let me get my writing utensil. You're going to see a picture of a cell, and you're going to see all water with arrows going in. Okay, something like that. In an isotonic solution, you'll have some arrows going in and other arrows going out. So it'll look something like this. Okay. Hypotonic. That's isotonic. In a hypotonic solution, it's your water is going to want to move out. So a good example of this is have you ever put salt on a snail? So a common solution to getting rid of snails is putting, or at least killing snails, is putting salt on a snail. What that does is it pulls all the water out of its tissues and kills it. Doesn't sound very nice way to go. So anyway. Those are the three things. Let's see, we got 4 minutes left in class. What else? Is there anything else I need to cover? I think I am there. Let me just really quick check another set of notes. Oh, active transport. I got to really quickly cover active transport. Active transport. Okay, so active transport, there is active facilitated transport. Okay, active meaning that it requires energy. So all active transports require energy. But it's facilitated in that it requires a transport protein. So there's special proteins that use energy to pull things in. So what does this mean? It means that it's going from an area of low concentration to high concentration. Okay. So if we got a cell membrane and we've got lots of solutes in there, they're gonna tend to want to go in this direction as far as passive transport. But what if you cannot have any of this solute inside the cell? Well, you're gonna want to pump these guys out, and that's gonna require energy, okay, to get those guys out because you're going from low to high. All right, so that's active facilitated transport, low to high. Really important to remember that. There are two other ways, but they're very similar in their process. They're called endocytosis and exocytosis. And I've got just 2 minutes to show you an example. What this is these are movements in or out of a cell. These are large molecules. Even whole cells. Okay. If you're going to bring in a whole cell, you're going to eat a whole cell or expel something out of a cell that's large. This is called endo and exocytosis. And so I'm going to show you a brief video on, so what I'm going to show you is an amoeba and a paramecium. Have you ever watched the movie Kindergarten Cop? oh, maybe it wasn't the kindergarten cop. Something about an amoeba. I can't remember now. Amoeba versus the paramecium. Let's cancel that. Come on. And I love the amoeba versus the paramecium. It's looking at, oh, let me bring up. Come on. All right, make this big. All right, this big goo cell here, it looks kind of weird. It's an amoeba, okay? It's a eukaryotic cell. You remember our 3 domains from Wednesday. Both of these are eukaryotic cells, so they both have a nucleus. But the amoeba is much bigger than the paramecium. And in fact, the amoeba likes to eat paramecium. So look what happens here. This is an example of endocytosis, and exocytosis would be just the opposite. So endocytosis, look what happens here. Get rid of the music. So the amoeba is headed towards the paramecium, not very fast, it's patient. All right. So the amoeba is now kind of caught the paramecium. Oh, it's gonna get away. No. So the amoeba is moving its membrane around the paramecium. Okay, this is endocytosis. Oh, it's caught. It is not going to get away now. It's trying to get away. Too late, bud. You're food. So these 2 one-cell organisms, pretty interesting that they can act in this way. And now what's going to happen is the paramecium is going to close off the gap and fully envelop the paramecium and slowly digest it. And so that's endocytosis. All right. If you have questions, otherwise, I'm going to cut you loose. Have a safe and happy weekend, and I'll talk to you later.